Okay, you guys, we are finishing up chapter 19, if I can pull this up for you. And we are finishing out talking about blood groups and blood types. What you may notice as you're working through your labs connected with this, and I hope everybody is okay in terms of signing into your HOL labs. If you're having a problem, let us know. One of the things that they do really, really well is the blood typing and the explanation for the blood typing. So don't just count on the PowerPoints to help you through this. The big thing to remember is that virtually every cell in the body has proteins sticking on the outside of its plasma membrane. And later on in this semester, you'll actually find out that when we get to immunology, our bodies are really, really good at recognizing self versus non-self proteins. Remember, your genetic code is a code for a protein product. Therefore, if your body recognizes something as foreign, it's going to attack it and make antibodies against it. And that is the very basis for these blood groupings. Now, your red blood cells are going to be marked by genetically determined glycoproteins and glycolipids on the outside, and they are called agglutinogens or isoantigens. And so when we look at that, we'll see that there are actually 24 different blood groups and 100 different antigens that we can actually take a look at. Because we're genetically controlled, we do have different blood types that vary among different populations. And that classification of your blood type is based on those proteins that are on the outside of your red blood cells, and we generally refer to them as A, B, AB, or O. O does not mean O as in LMNOP. It means zero, the absence of any protein sticking on the outside. Additionally, we're going to be taking a look at another antigen called the Rh factor. This antigen was actually discovered in the blood of rhesus monkeys. And so if you have that particular protein on the outside of your blood cell, you will then become Rh positive or Rh negative. So when you're looking at this particular one, when we are looking at that particular blood typing, if you will, Rh positive means you have the protein. If you are Rh negative, you do not have a protein. And the same is going to be true for any of these things here. If you are blood type A, you have protein A. Blood type B, you have protein B. Blood type AB, you have protein A and B. And with blood type O, you don't have any of those proteins. So as you can see, we have different blood types for different groups because they are genetically determined. Okay? You can see, for instance, that 79% of the Native American populations are O positive, whereas the European Americans, only 45% of them are O, and 85% of those are Rh positive. So because we have that protein or that antigen on the outside of those red blood cells, our blood plasma, the liquid portion of your blood, will contain antibodies that are going to react, okay, with A or B antigens. Okay, you are not going to have the antibody against your own blood type. Again, your bodies are really, really good at recognizing self versus non-self proteins. So a person with type A blood has the A antigen or protein on the surface of their red blood cells. That's what these little purple things are. But they will contain antibodies in their plasma against B. So if you get, so these people cannot accept anybody with a B antigen on it, okay? That means anybody with a type B or anybody with a type AB blood, because we have the antibodies to fight against it. Anyone that has blood type B, you have the B protein or antigen on your red blood cell. That means that you have anti-A antibodies, so you cannot accept, okay, anybody with a type A, okay, on it. So that means A and AB. Type AB people, they contain both of these proteins, so these guys can accept type A, type B, type AB, and type O. Because O isn't LMNOP, O is zero protein, okay? So these guys are the universal acceptors. People with type O don't have any antigen on that red blood cell, so they have both anti-A and anti-B antibodies in their blood plasma, so that they cannot accept anybody that has type A, type B, or type AB. They can only accept a type O. Therefore, type O people can give to everyone, so type Os are known as the universal donor, because they do not contain any protein for anybody to react to. So they're the universal donor. Type A people are the universal recipients. So this table, which is 19.6, really demonstrates the different blood types in terms of the antigen that's on their red blood cells versus what antibodies are in their plasma and what compatible donor types there is. Okay, so when we determine a person's blood type, we're going to cross-match, and you're actually going to do this in your own kitchen. If you follow that lab, you are going to take a drop of blood and mix it with serum that will agglutinate the blood cells that possess the agglutinogens that react with it. Agglutinate is a clumping. So when you see this, you're going to see kind of a clump in your wells. Okay, so when you take a drop of blood, okay, so this is going to be your drop of blood. You are going to need 3 drops of blood for each sera that you're testing. If you treat it with anti-serum A, this is what we mean by clumping, okay, or agglutination. Here, and here, and here, and here. Because anti-A is going to clump with A, etc. So if you're clumping with serum A, you will be blood type A. If you clump with anti-B serum, you will be B. If you clump with both of them, you will be AB. And if you don't clump with anybody, you will be type O. All right, there is something called the hemolytic disease of the newborn. This happens when an Rh negative mom and an Rh positive fetus occurs. And what happens is that there will be a mixing of blood at birth. What happens, the mom's body creates the Rh antibodies unless she receives a RhoGAM shot soon after the first delivery. The second, it only occurs with the second child because you're building up antibodies, and it does take a while to do that. We are looking, the shot that they generally get is called RhoGAM. After the first delivery. Or it doesn't even have to be a successful delivery. If there was a miscarriage or an abortion, she will need the RhoGAM shot for the next pregnancies, okay? So she's building up antibodies from the first pregnancy that will try to attack the second. So what we're looking at is transfusion and transfusion reactions when we are looking at blood types. A transfer of whole blood cells or plasma into the bloodstream of the recipient is generally used to treat things like anemias or even severe blood loss. An incompatible blood transfusion occurs when you get an antigen-antibody complex forming between the plasma antibodies and the foreign proteins that are on the donated red blood cells that will react with or cause agglutination. What happens is your donated red blood cells are going to become leaky and they're going to burst. And then the loose hemoglobin actually causes kidney damage. Problems caused by incompatibility between the donor's cells and recipient's plasma, okay, can also be a problem. All right, so let's look at some blood disorders. Blood disorders include things like sickle cell disease. It's a genetic anemia, which is the failure of oxygen carrying capacity of the blood. And they contain something called hemoglobin S that causes the red blood cells to bend into a sickle shape when it gives up the oxygen to the interstitial fluid. This is a change in 2 amino acids. Okay, remember when hemoglobin is a protein, you know it's a protein because it ends in IN. Remember, proteins are made up of chains of amino acids. So when you're looking at your whole definition of proteins, remember we did this way back in September. Proteins, we're looking at chains of amino acids. With sickle cell anemia, we are looking at a change in the sequence of 2 amino acids. if you remember back to last semester when we were talking about the compounds for life, the organic compounds for life, one of the things that we talked about when we were looking at proteins is that if you change the sequence of amino acids in your protein, you change the shape of your protein, and therefore you also alter the function of that protein. And that protein can be a wide range of problems dealing with inefficiency to perform or functionally dead. In this particular case, we've simply reduced the ability of that hemoglobin to carry sufficient oxygen to all areas of the body for them to perform their metabolic activities. Sickle-shaped cells also rupture very easily, and therefore they cause anemia and clots. A person with only one sickle cell gene, as opposed to two, has increased resistance to malaria. Okay, so we are looking at, when you're looking at genetically, what kinds of things gives you your traits, we are looking at 1 out of 2 genes here, or alleles, that actually gives the person the ability to fight off anemia, okay? So if you have only one sickle cell gene, means you have increased resistance to malaria. So this means that you're heterozygous for sickle cell anemia. And there's actually a picture of a sickle cell. All right, I want to do one more thing to talk about here. Let me end the show here. I want to add one more slide. Which it won't let me do, but at any rate, the other disorders that I wanted to stick in here that you're going to be able to see is hemophilia, so if I reduce that picture, we can see, we can talk about hemophilia, which is an inherited deficiency of clotting factors. When we looked at clotting factors, there were lots of them. Generally what happens is bleeding is going to be spontaneous or after minor traumas, nosebleeds, blood in urine, okay? There are a number of different hemophilias. Hemophilia A lacks factor 8. And we find that in males only. Hemophilia B, we're looking at lacking factor 9. That also is only in males. And hemophilia C is less severe, okay, because there is an alternate clotting factor activator. So this is the one that is less severe. Generally, treatment is transfusion of fresh plasma or concentrates of the missing clotting factor. The other disorder that I want to bring to your attention is leukemia. There is both two different kinds, acute and chronic. Since we have this prefix leuc, we are looking at white blood cells. So we have acute leukemia. Acute leukemia is an uncontrolled production of immature leukocytes. And so we have a crowding out of the normal red bone marrow cells by the production of immature white blood cells. So this is lots and lots and lots of immature white blood cells. And it actually prevents the production of red blood cells and platelets because of the crowding out. So this crowds out red blood cell production and platelets. Chronic leukemia is the accumulation of mature white blood cells in the bloodstream because they don't die, okay? It's generally classified by the type of white blood cell that is predominant, okay? So this chronic is mature white blood cells in the bloodstream. Okay, and generally it's classified by the type of white blood cell. For instance, monocytic or lymphocytic. Monocytic, remember, will break down and will mature into macrophages, and lymphocytic, we're looking at T cells and B cells. So I sort of wanted to cram that in for you. I really encourage you guys to pay attention to that lab. These things are step by step in your lab as far as blood typing is concerned. And I also, somewhere along the line, I had a blood typing interactive game that I'll try to upload as well. I have to remember where I put it. We'll try to get it up by the end of the first week. If you guys have any questions at all, please do not hesitate to ask us, email us.